anyone with obese, we attributed it to an unhealthy thing. But we also see fatty infiltration in the lean individuals. So what the earliest reports, of course, for the lean NAFLD have come from India. This was one of the papers that was discussed during one of the scientific sessions. Now, there are a lot of studies that looked at what is the association. If you see most of the papers from the Western world, they speak about PNPLA3, whether it is obese or whether it is lean, because that's the one that is driving the disease in the Western population. Now, there are a lot of genome-wide association studies and a lot of different markers that are already published. Of course, variants in HFP gene are also associated. There are few variants where if the waist circumference is normal, these variants don't produce any effect. Say for example, there's a gene called GCKR, and any individual with this GCKR variant, if the waist circumference is normal, there's no phenotype. But for some reason, the waist circumference goes to the higher side of the normal or even more than the normal, then it starts producing the phenotype, which in this case is fatty infiltration. So there are a lot of variants which have a lot of environmental interactions to produce the phenotype. Now, we did a study where we sequenced specifically again the complete exonic regions from specifically the lean NAFLD and identified a variant in a gene called phosphatidylcholine. This is a very interesting gene because it catalyzes the phosphatidylcholine is actually synthesized, processed by this particular gene. Now, in an animal study, when they removed the gene, the PAMT gene, the animal could not process this particular phosphatidylcholine and it required external choline supplementation. Now, whenever this external choline was not supplemented, there was fat in the liver that progressed very fast in the animal model. So this variant probably does not produce the protein. There's some problem with the choline synthesis, and therefore there is fatty infiltration, and these people are apparently lean. So what we hypothesize is probably if we can identify these individuals with the variants who are apparently lean and supplement them with choline, I think that would benefit them from progressing to the higher etiologies. Again, this is more specifically pronounced in the lean individuals. Now, usually what we see is more often than not there is a divide between the bench side and the well side. That's what we are trying to bridge. Now, we do the PNPLA3 genotyping and TM6SF2 genotyping that of course predicts the future onset of the disease, fatty infiltration in individuals. Now, we have identified the relevance of rare variants in this particular population, and of course it's not the PNPLA3 which is there, which drives the disease in this population. Probably the relevance of rare variants is shown in our population. Polygenic risk scores, again, which are relevant to this particular population is what we have identified, and we plan to put together a group of variants that have the ability to predict the future onset of the disease. PNPLA3 HSD splice variants, whoever has these variants and they have low VLDL levels, we feel that they would progress very fast, and these are the individuals that have to be identified very early, and appropriate nutrition and lifestyle based interventions have to be given to them. PMT gene, specifically in the lean, the variant carriers have to be supplemented with choline, probably that would benefit them. So this, as of now, is the translation that we were able to do. Of course, all this work is the PhD work by one of our students, Mr. Govardhan, who has now transformed to Dr. Govardhan. He was just awarded the PhD degree